Carlo is director of the Arizona Cancer and Evolution Center. He's also the first president of the International Society for Evolution, Ecology and Cancer. He was co-founder of and director of the Center for Evolution and Cancer at the UCSF. Carlo is very well known for his work on tumor evolution and on Peto's paradox, that is to say the puzzle of how large and long-lived organisms suppress cancer. And this morning the title of his talk is Leveraging Evolution and Ecology to Control Cancer. So thank you, Carlo.
Thank you very much. Since I'm the first speaker, I thought I should reinforce what Frederic said about the evolutionary nature of cancer before I talk about what I really want to talk about, which is how to treat it. So I think about cancer this way, that there are three necessary and sufficient conditions to get natural selection happening, and they are met in cancer. So one condition is you have to have variation in the population. There have to be differences. I think the phrase in French is vive la différence. There has to be differences between the individuals in the population. So if everybody's the same, there's no way for one individual to have an advantage over another. There's no way for natural selection to work. There has to be variation. That variation has to be heritable, or another way of thinking about this is there has to be some correlation between the parents and the offspring. If there's no correlation, then what was worked in the parents is not passed on to the offspring, and there's no chance for natural selection to have a grip. So I think of this as the traction part of natural selection. And then that variation has to affect the fitness of the individuals. So it has to change either the survival or reproduction of the individuals. And if you think about cancer, the cells in our body, they meet these three conditions. There's variation between cells in our body because of somatic mutations that happen in the cells during our lifetime. That variation is heritable, of course, because it's passed on to the daughter cells when a cell divides. And that variation does affect reproduction and survival. And you can see this in the hallmarks of cancer. Now, the hallmarks of cancer are phenotypes that are common across cancer for the very reason that they are selectively advantageous. So they keep evolving, re-evolving every time in each tumor. If you look at each of these phenotypes, they're an advantage that the cell would have over other cells that don't have the phenotype. So if a cell no longer responds to growth suppression signals, that cell has a reproductive advantage over the cells, its neighbors, that are responding to growth inhibitory signals. And you can go around the whole set of hallmarks. So this is what gives similarity across cancers. It's natural selection in our bodies. So I just wanted to lay that groundwork because all the talks this afternoon, I think, will essentially rest on that groundwork. But what I really wanted to talk about today is the fact that farmers are about 30 years ahead of us in oncology. And what Frederic mentioned, and I want to emphasize, is that our main problem in oncology, of course, is the evolution of acquired therapeutic resistance. So you apply a drug, it has a big selective pressure, you kill most of the cells, but some cells, by bad luck, have a mutation in them that make them resistant to the therapy. And that's true for all therapies that have been invented. It may be true for all therapies that will ever be invented. And so when the tumor grows back, over a period of months or years, it now grows back from those cells that were resistant. And so in general, when the tumor comes back, it is resistant to the therapy. And then you have to switch to another therapy. So this is the general problem we have in oncology, and the reason, in general, we haven't been able to cure it. Most of the cases where we can cure it are cases where there just aren't resistant cells there in the first place. So farmers actually have the same problem. So when they spray a field with pesticides to kill insects or pests in their fields, they select for resistance. Also, when they're trying to deal with weeds in their fields and use herbicides on weeds, they select for resistance. And about 50 years ago, they learned that if you just use monotherapy, a single pesticide, you spray a field, you kill most of the pests, but you select for resistant pests. Those resistant pests grow back and destroy the crops. It's the same problem we have in oncology. So about 30 years ago, they said, well, what if we combine drugs, right? If the chance of resistance to one drug is low, the chance of resistance to multiple drugs must be tiny, right? So maybe we can solve the problem by combining drugs. Doesn't work. They learned about 30 years ago that when you spray fields with multiple pesticides, you select for multi-drug resistance. The pests come back, they destroy your crops, end of the game. So they've addressed this problem, they've been learning from it, and they've come to something called integrated pest management. And I'm going to tell you about the lessons they've learned and how we might try to translate those to oncology. Okay, so where are we in oncology? Here's the data about multi-drug cocktails in oncology. It works in HIV, but in cancer, here's the data for lung cancer, 2 drugs versus 1 drug. This is a meta-analysis, and you can see that when you go from one drug to 2 drugs, you get about a 17% reduction in mortality. So that's significant and good, and it's typical in many cancers now to use multiple drugs. If you go from 2 drugs to 3 drugs, you have no effect. So in fact, the standard of care in lung cancer in general is a 2-drug therapy. It's a little dismaying, but you don't get much past that. And in general, it only buys you a matter of months. Now, one of the open questions for combining therapies is, should you combine them up front or should you put them sequentially? And there's only a few studies of this that I can find. And in general, sequential does worse than concurrent. And from an evolutionary biology point of view, you might think that if a cancer cell is faced with the problem of solving 1 drug at a time, it's an easier problem than solving 2. That's the initial inspiration for why we do cocktails in the first place, and works in HIV. The same is true for chemo and radiation, just 2 different therapies. Do them concurrently, you do better than if you do them sequentially. But this isn't really the important question. The important question is this: should you do them together and follow survival, or should you do one, follow for progression, and then use the other? And the question really is this time period longer than that time period? And that's generally hasn't been tested. Okay. I wanted to show you, one of the things I do is I teach a class about evolution of resistance across systems. So this is a summary of the literature I've been able to find about the mechanisms of resistance. There are many different ways of getting resistance to a therapy. So you can get mutations in the drug target that prevent the drug from binding. You can increase efflux pumps that just pump the drug out of the cell or the organism. You can amplify the target or overexpress the target, so you just swamp out the effect of the drug. You can some drugs are actually prodrugs and require a conversion into the active form in the body. And so one thing that cells can do is prevent that conversion of the prodrug. You can compensate for the action of the drug. And we see this in some cases by for instance, just upregulating the pathway. You can prevent uptake of the drug into the cell. You can destroy or inactivate the cell, the drug, for instance, with cytochromes and other detoxification agents or enzymes. You can actually protect the target, almost like put armor on the target. This is something we don't actually know if that happens in cancer or not. You can activate a downstream protein if the upstream is inactivated, and you can activate some bypass pathways. And you can see these are very common across all systems: malaria, bacteria, helminths, weeds. It's just this is evolution in action. There are many different ways of doing this. So unfortunately, that means there's also many different ways that cancers can escape our therapies. Here's an interesting bit of literature from the farmers, actually, with there's actually an herbicide, or fungicide, sorry, in resistance. And they looked at what factors affect the rate, the time until resistance evolves for fungicides. One thing that affects it is how many generations of the pathogen occur per year. So faster reproducing pathogens evolve faster, and they tend to get resistance faster. This is one that was most striking to me. If you look at the molecular complexity of the drug you're using, the more complex drugs actually have a shorter time to resistance than the simpler drugs. And it looks like the probability of binding decreases with the complexity. So you get very specific targeting with a complex drug, and that just actually makes it easy to mess up the drug with mutations in the target. If there are fewer host species for the pathogen, in this case the fungi, then you get faster resistance. And the idea is that if there's more hosts where there are essentially refuges for the pathogens, then the sensitive pathogens, in this case the sensitive fungi, are able to stay in the population and then come back into your fields. And this is actually, we're going to get back to this when we talk about adaptive therapy, and I think Joel Brown is going to talk a lot about the idea of preserving sensitive cells in the tumors. Similarly, if you protect crops in greenhouses so that there aren't fungi coming in from the outside, you get faster resistance. So somewhat counterintuitive. Okay. So one thing that pest managers and farmers in general do is they assume resistance is present. They assume it's there. And if you think a little bit about how we deal with cancer, we don't assume that resistance is there. We hope it's not there, and we go in with high-dose therapy and try to cure the tumor. They don't. They assume it's there, that going in with high-dose pesticides is not going to work. And they've evolved, sorry for the term, they have come to this approach called integrated pest management, which integrates a lot of different approaches to controlling resistance in pests. So the first thing they do is they really try to understand the ecology of the pest they're dealing with. And then they can approach the control of those pests through all the different channels of ecology, through the predators of the pests, through the pathogens of the pests, through the habitat that the pest best grows in. And then you can change those things. So you can bring in pathogens for your pests, you can bring in predators for your pests, a little bit like immunotherapy for cancer. You can change when you're planting, how much water you're using to try to change the habitat that benefits your crops over the pests, etc. The other key thing I wanted to actually focus on is this. They observe every day what is happening in their fields. They check their traps, they're looking at how their pests are responding to their interventions, and they're adjusting what they do. Now, how does that contrast to what we do in oncology? Typically in oncology, we have a set protocol. We will apply a chemotherapy once a week for 3 weeks, give a week off, do another cycle of 3 weeks. We do this for some prescribed period of time. We wait for a number of months, and then we check what happened. Months after we do the chemotherapy. It's a little bit like trying to fly a plane from Paris to New York, covering up all your instruments, covering up the windshields, and just following a set of instructions. You know, gun the engine for this amount of time, turn the steering wheel for this degree, gun the engines again, hope that you're on the runway at that point, gun the engines very hard, right, lift off. And then, is it any wonder that only about 10% of those planes arrive in New York? Right? It's very hard to do if you don't watch what's happening to with your interventions. We don't do that in cancer, and I think that's going to have to change in the future. Because no matter what we do, it's going to be responding to what we do. So they use a variety of ways of controlling. They mechanically remove pests, just physically removing them. They use biological controls, like I was saying, pests, pathogens and predators. And then, of course, they do use chemical controls. So these are our drugs. And they look at what happens, and they adjust, they think, they adapt, and they go back and they do this cycle. So what can we learn? So here are some of the principles from pest management. So they don't try to eradicate. They know if they go in with high-dose therapies, they will just select for resistance. They don't go for eradication. They assume resistance is there. They go for control. So they are willing to accept some level of damage as long as they can keep control over the pests. So they also use a combination of very different controls, as I was saying, cultural controls, that is like when you're watering, how you're fertilizing, mechanical, biological, and chemical. And they also don't treat if the pests are below some level. So they tolerate some level. And then they try to minimize their chemical interventions. And note, as you probably know about in the 60s and 70s, the environmentalist movement came up. There was the Silent Spring book about how we're killing all of the birds from our pesticides. And so they also are very much aware of the toxicity to their environment. And of course, we have the same problem in cancer, the toxicity to the normal cells in the body. They try to preserve it by minimizing their chemical interventions. And that gives them also less resistance. So why would you use low levels of chemotherapy in cancer? Here's the intuition. So this is something that Bob Gatenby and Joel and other people at the Moffitt Center have sort of shown us, that in general there's a cost to resistance. There's a fitness cost to resistance. In the absence of drug, sensitive cells will outcompete resistant cells. So you either have to run those efflux pumps, which Bob and Joel have measured. It uses approximately half of the entire ATP in a cell to run the efflux pumps to get resistance to doxorubicin in their MCF7 cells. So it's a huge cost. You might just have to change the protein from a optimal shape to a less optimal shape to avoid binding of the drug. In general, there seems to be a fitness cost to resistance, especially when there's limitations in resources. Of course, tumors are heterogeneous, so there's, as I said from the beginning, there's some resistant cells and some sensitive cells. And so we can use this competition between the resistant cells and the sensitive cells to try to control the resistant cells. So the central ideal here that comes from farmers is we need to preserve sensitive cells in order to use competition to control the resistant cells. And I think of it a little bit like this. So oncologists can use therapy to control the sensitive cells. The sensitive cells can use competition in the tumors to control the resistant cells. So it's a little bit like the enemies of our enemies are our friends. But what we do typically in chemotherapy is we go in with very high dose therapy, we kill all of the sensitive cells, and what's left are the resistant cells. Then we can't control them, they blow up, they kill our patients. So the idea behind adaptive therapy, in order to maintain sensitive cells, is to set up a negative feedback loop. So when you're killing too many of the sensitive cells, you back off on the therapy. If the tumor is growing too much, you have too many sensitive cells, you go higher with the therapy. So you just try to maintain this homeostasis, maintaining some sensitive cells that you use to control the resistant cells. And I'm going to leave, Joel, you're going to give the data on this, right? So I'm not going to steal any more thunder from you. Okay, so there are other principles from about chemotherapy from pest management that I'm interested in, and I think we should be thinking about and testing in oncology. One of the first ones is you should diversify the modes of action of your drugs. You shouldn't use two drugs that are killing the pest in the same way, or in this case, cancer in the same way. So I just pulled up the standard of care for lung cancer and pancreatic cancer for chemotherapy. They're both two drug therapies. Typically, there's a platinum drug for non-small cell lung cancer, plus some other chemotherapy, and there's more variation in what the other one is, but for instance, docetaxel. So here we have an alkylating agent that cross-links DNA and also alkylates the DNA, and we have another mode of action that inhibits mitosis via targeting the microtubules. So they're different modes of action, except they're both basically just targeting proliferating cells. Pancreatic cancer is worse, so typically it's gemcitabine and capecitabine, both of which are thymidylate synthase inhibitors. This is a prodrug, but this one's not. So in fact, they're using the same mode of action. Now here's a diagram of the different modes of actions for chemotherapies. So you might, the farmers would tell us, the agriculture researchers would tell us to diversify what we're using when we're combining drugs. This is a paper we did where we looked at the causes of extinction of species and asked, can we learn from extinction biology to drive a cancer extinct? But there are actually very many different approaches that we can use for controlling cancer, not just chemotherapies, but of course immunotherapies, bacteriolytic therapies, so using bacteria, using toxic forms of glucose, targeting the microenvironment of the tumor. Of course, surgical resection is one we use all the time. Maybe even slowing the evolution of tumors by targeting the mutation rate or the generation time of cells. Changing the habitat, for instance, just the temperature. All of these are very different modes of action that we should be exploring to see if we can do better. Another general principle from agriculture is to use the minimum possible dose. So this is completely opposite from what we do in oncology. We use the maximum tolerated dose in general in the cancer clinic. The farmers use the lowest possible dose. And here's some interesting results from non-small cell lung cancer. So they were doing a study actually to look at quality of life. So this is late stage metastatic lung cancer. There's a very low chance of getting a cure. So they tried, if we just go with a lower dose, can we improve quality of life? That was the main objective. Indeed, they can, of course, compared to standard of care with high dose therapy. Quality of life is better when you use lower doses. But the surprise here is that survival time is also significantly better when you use lower doses. That's an important hint. But has not been tested in most cancers. Okay. A third principle from integrated pest management is to try to partition your drugs either spatially, in their case spraying different fields, or in time, so spraying at different times. In fact, they say that you should never use the same drug twice in a row. Right? Compare that to what we do in oncology. We use the same drug over and over and over until it fails. Right? They will not use the same drug twice in a row. So how might we do this? We never really explored. Could you apply different drugs to different mets in the body? Could you use different regions? So also the work from Moffitt. Moffitt's a great place for cancer research, I have to say. Some of the work they've done is that you get very different ecologies on the periphery of the tumors versus in the center of the tumors, and so perhaps we should be targeting those areas differently. So there's a number of caveats, of course. I've been drawing this parallel between pest management and cancer biology. But of course, pests reproduce sexually. Cancer cells reproduce asexually, as far as we know. It's slightly an open question. We have an experiment in that. But in general, and this is important also because in general, resistance in pests is a recessive trait. So if you have sensitive pests that are breeding with resistant pests, then 3/4 of their offspring will be sensitive. So you can actually leverage that in pest management. We can't leverage that in cancer. They have growing seasons, so when they say don't use the same drug twice, what they mean is within the same season. So in the next season, they might go back to a drug they used in the previous season. We have limited cultural controls of how we're controlling cancer. And one that we're actually very interested in is switching from trying to cure cancer to trying to control cancer is a big psychological shift, right? When we have cancer, we want to get it out. We want to destroy it. We want to cure. We want to go back to the life we had before we had cancer, right? It's a little bit of a pipe dream because I have to tell you, as someone who was diagnosed with melanoma when I was 32, you never know if it's really cured, right? You will never go back to the life you had before. That being said, we would like to cure rather than control, but when we try for the cure, we shoot ourselves in the foot. We kill all the sensitive cells, we leave resistant cells, and it often kills the patient. So how do we go from going for the cure to going to control? And I think one of the things we can think about is the parallel with something like diabetes. We live with diabetes, right? We manage it. And so an image of the future is that we will be managing our cancers so that we live with them, but we will not be dying from them. One possibility for the future is that getting your cancer is something, just a stage of life. It's like being a grandparent, right? At some point, you will have your cancer, and then you will live with it, right? It doesn't have to kill us. It also requires a cultural shift in oncology, right, to go from trying to cure to trying to control. So one of the things we're launching is some psychological research and sociological research about how best to communicate, how best to talk about with patients and doctors this shift from cure to control. In fact, what Athena Activist describes this as cure by control, that we can shift our thinking about what a cure means. Okay, so here's those principles from pest management. Assume resistance is present, diversify your modes of action, limit the dose to the lowest practical level, and partition those modes of action in both space and time. This being said, these have not in general been tested in either mouse or humans, so we need to do the preclinical and the clinical trials to see if the lessons the farmers learned can be translated to the oncology clinic. Now, that's, I think, the set of ideas you can get from agriculture. There's another set of ideas that come from our community of just thinking about the evolution and ecology of cancers, and I just wanted to briefly review those at the end here. So if therapy selects for resistance, so if we're in the situation with a heterogeneous tumor, we select for resistant subclone and it grows back, then can we target these cells before we go in with the therapy? So we call this the sucker's gambit. Can you sucker this tumor into a vulnerable state by first applying some drug that specifically targets resistant cells that are going to be resistant to your chemotherapy, and then you sucker the tumor into a vulnerable tumor that then can be cured by your therapy? Another idea from Bob Gatenby and colleagues is an idea of what they call double bind therapy. Can you use drugs that select for diametrically opposed types of resistance so that there's actually antagonism between the kinds of resistance that the two drugs apply? And if you could do that, then when you apply both drugs, there should be very difficult to get multi-drug resistance. Another idea is to ask, well, what would you like to select for? And this is something that Frederic was just pointing out. We can drive the tumor. Once we understand it's going to evolve to whatever we do, we should be asking, what should we do? What are the things we want to select for? Right? And one answer to that question is benign cells. Can we select for benign cells and against aggressive cells? And we call this the benign cell booster idea. Can you increase the fitness of benign cells, not just kill the more aggressive cells? And so we did some simulations. This is an example of simulation where these are the benign cells. Over time, they get outcompeted by the more aggressive cells. You come in with a benign cell booster at this point, and you can get the benign cells to outcompete the aggressive cells. Okay. So this is the end of my talk. I'm not sure how I'm doing for time, but I think I'm a little bit ahead. Okay. Hopefully I have a problem. I talk too fast and I mumble, so I apologize for that, especially being in France. But the underlying idea here is that cancer is an evolving disease. The cells are evolving by natural selection. And so we need to be using evolutionary theory and ecology to think about how to control and treat this evolving disease. And I want to acknowledge the grant support and all the wonderful collaborators I have, many of whom are in the room. Thank you very much.